Okay. All right. Well, hello and welcome, everyone. Thank you for joining us tonight. My name is Andrea Dickinson. I'm on the board of Sustainable Plymouth, and I'm also on the Town of Plymouth's Open Space Committee. As I'm sure many of you recall, late summer and early fall of last year brought concerns regarding Eastern equine encephalitis, or EEE, to the forefront in Plymouth as the state flagged our community as high risk, with the first animal case of EEE in Massachusetts in 2024. Public fields and parks were closed dawn to dusk, and outdoor events were moved inside. Aerial spraying was underway to help curb the spread. Meanwhile, a lot of folks were left scrambling with questions about impact of spraying on their backyard crops, beehives, wildflower meadows, and potential impact on our pollinator populations. Questions about possible exclusion zones and whether or not to cover their crops and or beehives arose, and we hope tonight's forum will help answer some of those questions and more. I want to thank our esteemed panel for taking time out of their busy schedules to speak to us tonight. They bring a wealth of knowledge and expertise to help us better understand EEE, mosquito spraying, and the impact on our local environment, specifically on our local pollinator populations. We'll start with a brief presentation from each of our panelists before moving on to a Q&A. So we'll be starting with Dr. Catherine Brown, State Epidemiologist and State Public Health Veterinarian. We'll move on to Ellen Bidlack, Entomologist with Plymouth County Mosquito Control Project, and Blake Dinius, Entomologist Educator with Plymouth County, will be wrapping up the presentations. So we'll move on to Q&A, and then before we close, Sarah McCormick with the Town of Plymouth, Natural Resources and Sustainability Specialist, will touch on the Town of Plymouth's Climate Action and Adaptation Plan as it relates to vector-borne diseases and climate health risks. So without further ado, Dr. Brown.
All right. Good evening, everyone. I know some of you, but not everyone. I usually go by Katie. I'm relatively informal. So please feel free when we get to the Q&A to call me that. So I've been with the Department of Public Health for 19 years now. And in my previous life, I was actually a wildlife veterinarian. I worked on Cape Cod and then actually out in the Midwest as well. So I come from environmental wildlife roots and ended up in public health through a long, complicated process. Anyway, I just want to talk a little bit about why EEE is taken so seriously in Massachusetts. Probably some of you know this. So I'm not going to try to make you experts in one night, but hopefully just enough to be able to sort of understand where the Department of Public Health is coming from. So I'm going to talk about a couple of different things. So just an overview of West Nile virus and EEE, the seasonal cycle of EEE in the environment, what the department's role is in EEE surveillance and response, our prevention tools, because I wouldn't be doing my public health job if I didn't mention those, and then a little bit about is EEE increasing in Massachusetts. So I know this is a little small, but essentially this is just a comparison contrast between the two mosquito-borne diseases primary mosquito-borne diseases that we have in Massachusetts. West Nile virus, which is actually the one that kind of occurs every single year, right? Even if people are not familiar with EEE, they're usually familiar with West Nile virus. You get exposed through the bite of an infected mosquito. Most people exposed to West Nile virus will never get sick. They'll never know they were exposed, which is good news for them. Only about 1% of people who are exposed will have a relatively severe form of the disease where the virus gets into the central nervous system, the brain or the spinal cord. And this is something where the older you are, unfortunately, the more likely you are to have serious disease. EEE, by contrast, is less common. It doesn't occur every year yet. Let's hope it stays that way. But we actually think that most people who are exposed develop very severe disease. So it doesn't have that asymptomatic infection, we think, that West Nile virus does. And we see in Massachusetts over time between 30 to 50% mortality rate from cases, and then 80% of people who survive are left with some level of permanent neurologic disability. So if you have to choose one disease to get, pick West Nile virus. All right. I shouldn't joke about this. It is serious. I think it's important to understand a little bit about how this cycle works because it also helps explain how control is conducted. And my esteemed colleague will take over more of that in the next presentation. But so what we think happens is that birds that are carrying the EEE virus come up from the south in the spring. We often blame Florida. They actually have viruses with the same genetic kind of fingerprint as ours. So gets introduced into Massachusetts. So there's some number of birds carrying the virus as they come up. There are certain mosquito species that live in swamps in white cedar, red maple, hardwood swamps that we have a lot of here in the southeast Massachusetts. The birds end up in those swamps. The mosquitoes bite the birds, pick up the virus, and then go and bite another bird and are capable of spreading the virus to them. So you can see as even if only a few birds are bringing the virus in, as you have more and more mosquitoes, they are biting the birds, picking up the virus, spreading it to new birds who are then bitten by other mosquitoes who pick up the virus and spread it to more. So this is called our amplification cycle, and we just end up with more and more virus. I say in the environment, but it's always actually in the mosquitoes or in the birds. And then at a certain point, and this, the time point, it's not kind of set in stone. We can't actually detect it immediately. But at a certain point, there will be enough birds that are infected that an entirely new species of mosquito, different species of mosquito, will bite a bird, pick up the virus, and then bite a human or a horse or another susceptible mammal. So humans, horses, that second mosquito, none of them are really part of the natural cycle of EEE. We're all considered sort of spillover. And you can see on this graphic that in general, the amplification cycle kind of starts before June. It's going to go probably into at least August. And then we move into what we call the spillover cycle, where we start to see possibility of infection in mammals. And so it's really only during certain parts of this cycle where we can intervene with mosquito control. And unfortunately, when it comes to EEE, because of the habitats that are involved, trying to do larviciding or kill juvenile mosquitoes before they start flying and really become a problem is pretty much impossible because of the red maple white cedar swamps where the amplification mosquito lives. So what that leaves us with, unfortunately, is primarily adult mosquito control. All right. So the department, we work with mosquito control districts on mosquito collection and testing to do surveillance for the presence of virus in mosquitoes. We also, and our laboratory does that. The laboratory also tests all of our human specimens, people who are suspected of having either EEE or West Nile virus. And we also test some of the veterinary specimens. All of that is data for us, and it helps us understand sort of how much EEE activity we're having during the current year. But that's not really useful if we don't then take that information, kind of analyze it, and turn it into a risk assessment for all of you. So when we talked to Plymouth over the last summer, I don't remember what day it was anymore, but anyway, when we did and said, hey, there's a horse that lives in Plymouth that was infected there, right? We consider you now to be at this level of risk for EEE, right? So we take the information, the data, and turn it into risk communication. And then we also need to use it to help people understand where and when specifically the risk is occurring. One of the primary ways we communicate that is through our risk maps, which are updated kind of daily throughout the mosquito season as the data changes our risk assessment. And the colors are used to sort of tell us, are we at remote risk, are we at medium risk, or are we at critical risk? And just as, oops, sorry, I thought I had another thing. I will just tell you that each of the colors means something different, and when you hover over it, on the map, it will pop up and tell you exactly what it means. All right, so prevention tools. I've already told you we don't really have larviciding as a good prevention tool for EEE. The picture doesn't do it justice, but these are actually the little kind of crypts, the holes in the ground where the juvenile mosquitoes live and breed. And it's really hard to get any type of product in there to be able to reduce that juvenile population of mosquitoes. So larviciding is really not something we can use against EEE, although it's very effective for different species of mosquitoes, such as those that spread West Nile virus. At the Department of Public Health, our primary focus is on telling people where and when risk is happening and encouraging them to use personal protection, right? That is our focus. No matter what else is happening, even if we haven't found a lot of virus, you will hear me telling you during the summer that you should be, paying attention to the maps, using mosquito repellent when you're outdoor, clothing to cover up. And during some of the highest risk periods, we also make a recommendation about considering rescheduling your outdoor time so that you're not outdoor at the same time the most dangerous mosquitoes are. And then as risk starts to build and increase throughout the season, we rely so much on the mosquito control districts to help us control some of those populations of mosquitoes. And sometimes when risk is continuing to build and we are not seeing it really decline, and there are, risk for additional human cases, then we have to get into something like aerial spraying. So just to let you know, we have an educational website. We do try to refresh the materials pretty frequently so that people don't get bored of seeing the same picture all the time. We definitely purchase media assets during the summer to kind of depending on how much risk there is to help remind people about what's happening. And we do specific press releases at specific events during the season as well. So this is just an example of some of the data that is generated by the mosquito surveillance and also the human surveillance. So this is from the 1970s till 2024. The blue bars are human cases, number of human cases in a year, and the red line is the number of positive mosquito samples that we have in a current year. And you can see that they go up and down relatively well together. It's not a perfect match, but it does mosquitoes give us a pretty good idea of how much we need to worry about human cases. What I want to flag for you is that when I started at the department 19 years ago, the mantra was, well, we had outbreaks that lasted 2 to 3 years, and then there would be like 5 to 17 years in between cycles, and we no longer see that, okay? And that's what this graph, what I'm trying to show you. You can see that from 2004 to the present, that we are seeing increasing occurrence of some of these worse outbreaks. I will also say that you can see this is all EEE cases in the US, and you can actually tell that there's a little bit of an increase as well. The original slide kind of had a dotted line, which was statistically showing you that there is an increase over time. So it's not just in Massachusetts, but it is definitely partly in Massachusetts. And in addition to increasing kind of frequency of those outbreak years, we are also seeing spread of the virus into other New England states and into parts of Massachusetts that have not historically been at significant risk. So again, when I started, way back in the early 2000s, Massachusetts was really, and New York were kind of the places that had the most EEE cases. Since that time, we have now seen EEE cases in New Hampshire, Vermont, Maine, Connecticut, and additional parts of Massachusetts. And so my last slide is just to say that I'm afraid that that is probably going to continue, right? I think we are seeing some effects that are related to, one, climate change, and also changes in land use patterns, right? We have more people living in swampy areas. I don't really mean that, but down around green space, right? Because we like green space. I don't want to live in Boston. I don't know about you guys. So those changes in land use patterns and the fact that we are seeing climate change, increased precipitation events, higher temperatures longer in the summer, and then also alterations in songbird populations. And then last but not least, we actually are seeing some expansion of other mosquito species, invasive mosquito species, that maybe eventually over time could change what we see in terms of human disease in Massachusetts. All right, turn it over to Ellen.
Hi, my name's Ellen Bidlack, and I'm the entomologist at Plymouth County. I've been there for about 25 years, so I've been there a while. Definitely have seen quite a few changes when we're talking about Tripoli and West Nile and that kind of thing. So I'm going to explain a little bit more first about the mosquito life cycle because it's really important if you're going to understand our mosquito control strategies to make sure that we're all on the same page and understanding how the life cycle works. In the state of Massachusetts, there are 52 species of mosquitoes. Worldwide, there's about 3,500. And these species all vary a little bit in terms of what kind of habitat are they using, how do they overwinter, what kind of species are they attracted to, how many generations do they have every year. And so these variations, of course, allow them to separate ecologically. And the result is that they will play different roles when we're talking about Tripoli transmission or any other disease transmission. Some mosquitoes are going to be good vectors for one disease. Some mosquitoes might be good vectors for another, and others might never be involved with transmission of a mosquito-borne disease. So we have the adult female mosquito. She will only females bite. They bite in order to get blood so that they can have nutrition to develop their eggs. The males actually feed on nectar only. Females will also take nectar. She will lay her eggs, and she will lay her eggs in a variety of ways. This is a picture of some egg rafts, and each egg raft contains probably about 150 mosquitoes. Other mosquitoes will lay their eggs on the edges of the puddle. So that way, when an environment like a floodplain or something like that happens, those eggs will then be triggered to hatch. A lot of mosquitoes will overwinter as an egg, and in this case, with the ones with the rafts, those are usually like container breeding mosquitoes, that kind of thing, and they'll have many generations a year. So the larvae hatch out. The larvae are all the larvae are filter feeders generally, and so they're feeding on bacteria and organic material in the water. The life cycle, how long they stay in a larval stage, depends on the species. They could be a week, it could be half the year if they're overwintering as a larva. And then they will pupate. There's no mosquitoes in Massachusetts that overwinter as a pupa. The pupa stage will last a couple of days, probably like 4 days. This is the transition stage between the adult and the larva, and this is where the adult develops. And you can actually see the eye of the adult in this picture. On the bottom portion of the pupa case will split across the back, and then the female or the male will come out. Males will come out of the pupal case, will pupate first and come out. They take a couple of days to mature. They actually, when they emerge, their genitalia are upside down, so they have to rotate. They have to have time to mature in order to be able to mate. They will mate with the females. The females only mate once, and then they'll go out and host seek on whatever they're attracted to. It might be a bird, it might be mammals, it might be small mammals versus large mammals, it might be reptiles and amphibians, and there's even a few, I believe, that feed on invertebrates as well. So and there's some that don't blood feed at all. So a wide variety. we do what's called an IPM approach, or integrated pest management, or a tiered approach to our mosquito control. So the idea is that we're going to spend most of our time doing the things that have the least environmental impact. And so we have education and outreach. That's a picture of Erin. She does our primarily the education and outreach. She is she's our community liaison, and she's the only person in the state. We're the only district that has a community liaison that's specifically hired to do community outreach. We also, this is my portion of the project, is the disease and mosquito monitoring. We also do habitat reduction, we do the larviciding, and of course we do some adulticiding. So with the surveillance, I have to run a bunch of different kinds of traps in order to catch different mosquitoes, right? Because mosquitoes have different life cycles, I need different traps. So these are just pictures of the different traps. The main one I use for EEE is the CDC light trap, which is in the center, and it's just a light with some CO2 and a fan. And that's what I use primarily for EEE surveillance. And then the Gravitrap is mimicking a container, which is what we use to surveil for West Nile virus. So then, of course, we've got habitat reduction. So we can do a variety of things. We can do cleaning up. We're generally trying to free like roadside ditches and that kind of thing, and make sure that things are not blocked. So we're alleviating flooding and that kind of thing. We do not dig any new ditches. That used to occur, but we don't do anything like that. It's just strictly maintenance on existing infrastructure. So last year, to give you an idea of the scale, we did about 20 miles of ditch maintenance in the county this last year. And then we also do some tire removal. Each resident or each household can have us take away 10 tires for free. So you can give us a call in the fall or the winter to do that. Last year, we removed about 1,500 tires from the environment. And that's a picture of us. We took away about 7,000 tires, I believe, out of Norwell one time. So that's the picture of that project. So the larviciding is obviously, we spend a lot of time doing the larviciding. The goal is to do more larviciding than adulticiding. So the reason is that the pesticides we can use for our larvicide program are mostly biologicals, and we can treat a smaller area, and then the mosquitoes will never emerge to bite anybody, never emerge to transmit a disease. And so that's our preferred pesticide category that we would use. And of course, we have the adulticiding. We do only truck-mounted adulticiding. This is what's called a ULV, or ultra low volume sprayer. It basically turns the chemical into a very fine mist, and it can travel for about 300 feet from the back of the truck. We use about 1 ounce per acre. The rate depends a little bit on the chemical. Sometimes it's 0.6 ounces per acre. But so that's how we get to that low rate, is by really making very small droplets. The goal is to treat the air column and kill the mosquitoes that are flying and host seeking at the time that we spray. So we have a bunch of regulations and policies that are impact the things that we do and our decision process. So Katie already talked a little bit about the surveillance and response plan. And then we also are in communication with Natural Heritage and Endangered Species Program. And we also have a bee policy, and we maintain a pesticide exclusion list. So if you wanted to add your property to the pesticide exclusion list, you can do that. So let me talk a little bit about this. I won't go into too much about this. The surveillance and response plan is what DPH uses to communicate to everybody. It's their plan about how are we supposed to respond to diseases. Again, it's a tiered approach. So here is the example. You already saw the map, and there is a table, and it has a column, it says like triple E moderate risk. And then this defines defines what moderate risk is, and then has a list of actions for us. And those actions are things for us, things for the health department, things for people to do, that kind of thing. That's where the recommendations like to close ball fields are coming from. So they're on that surveillance and response plan. So natural heritage and endangered species, they review everything we do, whether it's tire removal, whether it's hand cleaning a ditch, whether it's adulticiding or larviciding, they review all our products and all our activities and give us maps and tell us where we can and cannot do certain activities. They'll even tell us like, yeah, don't go in here and pull out tires until you talk to us. So there are areas like that. So all our staff have a tablet with them that communicates to them where those areas are and what they can and cannot do in that area. So when you see, so for example, with that the air spray that happened last year, the map that you saw that was a very generalized map. And within that map was about 62,000 acres of excluded areas for endangered species and for our public drinking water. So we do have a bee policy as well, and the bee policy has been in place since 2007. And so the bee policy is written by the folks who regulate mosquito control in the state. It's called the State Reclamation and Mosquito Control Board. And they have basically set a policy for us that we can only adulticide 1/2 hour after sunset to 1/2 hour before sunrise. So we're trying to avoid the time when bees are most active. If you want to read more about the bee policy, it's on our webpage. You can also add your property to an exclusion list. The exclusion list is maintained by the Department of Ag, and they then send us an email when you've added your property. The list updates every year, so you have to put your property on every year. And then ideally, you're marking your property so we can see it in the dark, which is why we ask that you use something large and preferably a white pie plate, but no one ever does that. But like a big paper plate or something like that we can see in the dark, and we can know. If you just put up reflectors, it could be for any reason, right? So we ask that people do that. And then if you have any questions or anything from me that you need, this is my contact information, and there's lots of information on our webpage. It's plymouthmosquito.org. You can find out what pesticides we use. You can find out where our spray trucks are going to be. You can find the link to put yourself on the exclusion list if that's what you want to do. Yeah, you can find out just tons of information from there. Thank you.
Thank you. All right. Good evening, everyone. My name is Blake Dinius. I know some of you, and some of you are fresh faces. I work as the county entomologist educator, and I know I don't have a ton of time, but I do want to talk a little bit about how I kind of came into this particular role and my background, because I think it maybe helps introduce why I'm going to speak on the next topic, which is going to be about pesticides and ecotox testing. So as a kid, I grew up in like the 90s, and what was really big then was a movie and a show that kind of got me really into this. And the movie was called Fern Gully, and the show was called Captain Planet. And then I had a biology teacher who, I know Doug Tallamy is a really big name these days, but the name that was really big for me back in the 90s was Rachel Carson. I read Silent Spring. And so that kind of got me on this track where I really wanted to do something to really assist the environment. And I really wanted to help, and I wanted to make a difference. And I got into this field called ecotoxicology. And so one of the big things that kind of comes up, I think a lot, at least in communication with people and the general public, is that this notion of chemicals either being good or being bad, and then there's all natural chemicals that are supposed to be good, and there's synthetic ones that are automatically bad because some guy in a lab created them. And there's a lot of miscommunication, a lot of misinformation that kind of cycles that. And I wanted to kind of open up some of this black box that goes into the pesticide registration process, because it's something that I didn't actually understand before I got into this field. And once I got into the field, I was like, wow, this is really complex and really nuanced. And there's actually a lot of stuff that goes into it, but that the system is not completely perfect because it's a science, right? And it's not meant to be perfect from the get-go. It builds and we learn new things and then we change the process and then we add new things and then we change it again. And there's all these iterations that we're going about. How are pesticides registered? And that's actually a real photo. That's a photo I took of a beehive that I was actually grafting honeybees from for a bee study. So one of the things that kind of comes up in the world of ecotox is that it's the dose that makes the poison. So there's this concept that almost anything that you consume or you touch or you interact with can be toxic at some point. For instance, like 6 liters of water within, I believe, about 3 hours is toxic to a person. It can kill someone. You've got caffeine, and this one says 118 cups, but I actually did the math, and it's really about 105 cups of coffee, but the water actually kills you faster. The amount of water in that caffeine kills you faster than, but the amount of caffeine in it, just caffeine itself, is toxic at a certain level, but it's fine, and I'm running on caffeine right now, it's fine with just a cup, right? Just a cup, right? And then alcohol we know is toxic, it's a poison. And this is 13 shots is about someone about 150 pounds, gives you about a 40% alcohol, blood alcohol level. So like they can be, there's one of these things that they can be safe, right? If you consume just a little bit and then they're toxic at a certain threshold. So that is a really important concept when it comes to pesticide registration, because you're trying to strike that balance between what is going to be effective at a certain level and not toxic to other things. And you're trying to pick these levels in the environment and what you expect is going to happen in the environment when you introduce that product into that particular environment. And so you need a lot of data to try to guess, really estimate what's going to happen. And it's going to be based on a lot of different factors. One of the biggest kind of misconceptions is that the EPA, the Environmental Protection Agency, is the agency that runs these tests. And they don't. They don't run these tests to determine whether or not these pesticides are okay in the environment. They actually contract out this work to third-party labs. And then I have that building here. So if you are a pesticide manufacturer, you could be Bayer, Syngenta, you could be Novartis, you could be all these different companies, right? And you could be producing either a medicine or a chemical, and it's going to somehow, if it's a medicine, it can, like birth control, it can get introduced in our waterways. If it's a pesticide, it can get sprayed. You can't just put that in the environment either. You've got to register that with the EPA, and you get this testing done by this third-party laboratory that is in no relation to these. So one of the big things is that I get all this kind of, some people are like, oh, well, the EPA is in the pockets of this company. They're buying them out. They're paying them money. They're giving them all this money. That really dynamic can't really work because you have this third party doing that testing independently on their own, and they're doing it the best way possible. And they're actually catching things that the company itself, like if it was up to the company, they may push out a product, right? But you have this third-party company that their sole responsibility is to make sure that that doesn't make it out into the environment. And then this company, this laboratory, despite what that company wants, they give their data to the EPA, and the EPA makes that decision, right? So even if it's bad, even if it's data that makes that pesticide look bad, they still are obligated by law to give that data to the EPA, right? So there are guidelines that determine what these laboratories can, like how they run their tests. And this is very rigorous, and it's very, like if you run a test in one lab in Ohio, it's got to be the same exact test that you run in Massachusetts or in China or in Russia or in California, and they run it according to these standardized sets of guidelines. And it is like incredibly strict. Like it is like almost like laughably strict. So like one of the things, for instance, would come up is someone said, I started this test at noon. You had to record in a notebook exactly 12. If it's 12:01, that study gets thrown out and dismissed, and you can't run it. It's not a study that you can qualify as legitimate data, right? You can't round. If you put in exactly this amount of grams into the solution, it has to be exactly that amount. And if someone said they wanted to start it again 6 hours later, you can't start at 6 hours and 5 minutes later, exactly 6 hours later. And all of that has to be recorded in a data book. It has to be completely tracked and traceable so that someone 50 years from now can pick up that data book and replicate that exact study and get the same exact results without question, right? They will get the same data points, same exact results, and it is like I said, laughably crazy. It's like almost crazy, and you have people that their job is just to check the data book, and that's all they do is they watch over it and they just check the data book to make sure that you're filling these things out exactly. So it's crazy, and there's audits that kind of go on. There's a lot that kind of goes into it. And then, like I said, the endpoints that we're looking for and the data points that we're looking for are very complicated, right? So you have like a fate department. It's like they determine like how does this end up in the environment, right? So that's their goal. You have metabolism, like how is that chemical broken down? You've got acute toxicity, chronic toxicity. They've varied test organisms. Like for instance, I worked a lot with bees. I worked a lot with springtails, earthworms, a lot of inverts. We called ourselves the bug lab, but we were really just working. The only real bugs that we were really working with were actually like the bees. Like the springtails are technically not insects. And then you've got the lab, and then you got the semi-field and field. You've got a lot of different levels of testing, and each one of those has their own set of rules and is determined by a certain set of criteria whether or not you run that test. For instance, like our earthworm studies were basically triggered if the chemical had a certain property where it would bind very tightly to soil and could therefore stick in the soil layer, and then that exposure route would be directly to the earthworms and springtails. So you're not going to run these soil studies if something can pass through soil very quickly. You might run an aquatic study because that's where it's going to end up and that's how it's going to break down in the environment. And the endpoints we're looking for are usually calculated values. So one of the things, it's really hard to teach this in like just 15 minutes, but you're looking for these calculated data points. So like when you're running your acute study, what you're really looking to calculate is this thing called the LD50, right? And that is the dose, right, that you're going to kill about 50% of that test organism, right? So if you look at this, so when you have a very low LD50, that means a small amount of it can kill a very large, like it can kill 50% of that population, right? And so you calculate these values, and the LD50 is just an example because it's just one data point. Like you're also looking for no effect level. What is the level that there is no effect? Or what is the level of the lowest observed effect? You're looking for all these different things with the different variety of tests, right? And so all that data, molecular structure, all this stuff, that is the data that gets sent out to EPA. One of the other things that's kind of a secret, that I don't know if I'm supposed to tell you, but I'm going to say it anyways, is that the stats that we run on these are pretty much, you run your own in-house stats, but then EPA will also take that data in-house and they'll rerun all of the stats on that data as well. So even if you wanted to like skew the stats, which you wouldn't, you can't because EPA is going to run their own stats in addition to yours on their own. And they typically run this one stats thing called Williams, which is typically a more conservative, it gives you a more conservative value. So it's going to generally be a low, you're going to see like a chemical is going to appear even more toxic than if you were to run a less conservative stats method. And so all of this information, this is how they generate things like the label on a pesticide, right? So they take all of that information and they determine where it's going to end up, how it's going to break down, where you're going to spray it, what organisms are going to be affected. And then you get this label on the back that tells you what a person should spray and where they should spray it. That's why we say the label's the law, because if you follow that, you're following it not because it's a willy-nilly thing, but because specifically if you follow this, the EPA has determined that that's going to minimize the amount of risk that it's going to happen. It doesn't mean no risk, but it's going to minimize it based on the data that was run in those studies, right? So they're all basing it off of that data in those particular studies, right? One of the things I do want to bring up is Anvil 10+10, which is the chemical that a lot of people use in the adult deciding. And this is one of the things, so this came up in a Massasoit Community College poster, which is really cool. And they actually found that after the last set of aerial sprays, they said unexpectedly increased bee abundance was observed post-insecticide application compared to the time points with non-spray years. So they actually saw more bees in the years that there were sprays than, and to me that is not surprising at all because of the fact how I've worked with these chemicals. Like I said, we run these rigorous studies that are replicable, And then we send that data to EPA, and the EPA generates. So as long as you're following these labels, in theory, should cause no impact on bees because they've taken the level, a no effect level of bees, right? Let's say you've got your no effect level where you don't observe any effects on bees. EPA will drop that typically below that threshold, and that's the level that's usually being sprayed. So when they see no effect, it's below the no effect level. So you would typically usually see effects if someone's doing off-label usage or spraying too much, spraying double, they mix it improperly, all of these mistakes happen, right? So why do we hear about bad things about products? Like for instance, like this says, the main ingredient in Roundup doesn't just kill plants, it harms beetles too. Dun, dun, dun. But like it's typically misuse, off-label use, counterfeit products, improper mixing, the expected environmental concentration for some reason ends up higher than we initially anticipated. And then the last one is, I think, the one that tends to come up and is the most frustrating of all is the unanticipated consequences in novel products. So basically, so like my particular role in ecotox, I got into ecotox right around the time neonics were getting big, like right around 2009, we were talking about colony collapse disorder. And one of the big things that I did in ecotox was help develop the methods and guidelines that we now use to help monitor things like long-term chronic effects in bees. We developed like things like larval bee assays and long-term adult studies, and those were actually incorporated into the current suite of testing that is required for bees. But before that point, we didn't actually anticipate these things happening. Like before, it was typically acute effects. Does it kill a bee or doesn't kill a bee, right? And then if it doesn't, then we would generate our endpoints. And then the neonics, what they tend to cause are these very long-term chronic effects where they make bees. We observe things like they almost look drunk, like they bounce around their cages and they're not able to really act properly, but they don't die. They don't die. They stay alive, right? And so then that's how we would generate these no effect. That's why we would run these chronic studies, and that's where the importance of these chronic studies kind of came up. So again, before the 2000s, you typically just had these bee studies, the oral acute, the contact acute, and they were short studies, and the bees were dosed once. And then now, because of the advent of neonics, we have all these other studies that we've been adding on, but all of that methodology and all that development, it takes a long time. There's a long lag time before when you understand what's happening to when you develop and you basically, you get these guidelines and you get these tests put in place. So sometimes people will say like, oh, the EPA is doing nothing. They're doing something. It's just you can't just put something out there without validating it, without understanding if it actually is going to capture the data that you need. And so right now, like solitary bees, when I got out of ecotox, solitary bees were like the big thing. And it's just now last year that we've started to really validate these solitary bee studies, right? I was working with them a little bit back in like 2015, 2016, but now you think 10 years later, that's when we finally are starting to get the information to actually run these studies. It takes a long time to make sure you get those methods complete and that it actually makes sense and then you're going to get data. Because remember, if you run one study in Ohio, you've got to run the same study in Massachusetts, you got to run the same study in China, and they all have to work the same and generate the same endpoints. You've got these larger field and tunnel studies. I wasn't really involved in the development. I did a lot of stuff on a smaller scale. So at the end, I'll take any questions. I'm done.
So thank you very much to Kate, Ellen, and Blake for that presentation. So we're going to transition. Is this on? Is this on? Transition to Q&A.
The question I have around pollinators, you addressed bees, but what about your moths and butterflies? Are they also affected or have an increased population after this, or are they more affected?
So that particular study at Massachusetts, they weren't looking at the butterflies and the moths, but at least from what I've seen in other sets of data, again, as long as you're kind of doing it according to the label, that risk should be minimized. And I don't know if you and Katie and Ellen want to speak on how you kind of minimize that risk. spraying at night.
Yeah, I guess I'm not a toxicologist, so this is a little bit out of my realm. But yeah, I mean, when you're spraying at night, so that does help reduce contact with butterflies and things that are out in the day. Do you have anything, Katie, you want to?
Yeah, I mean, the other thing I've learned over the years is that one of the most important things is Ellen talked about how the product is sprayed as an ultra low volume mist. And part of the point of that is that so the droplet is sized appropriately to carry the right amount of pesticide to kill what it's trying to kill, which is the mosquito. Now, I'm not going to say like it's 100% and there's, nothing else that it could kill. But if you think about a droplet that has the right amount of chemical to kill a mosquito, and you compare that to something that's larger, like a butterfly or a dragonfly, that is one of the things that I think is supposed to help protect some of these non-target species, that the amount of the product is going to be smaller for the larger species.
Thank you very much. So if anybody has questions, feel free to step up to the microphone. We just ask if you could maybe keep your question to like 2 minutes or less, unless, for other people. Oh, and there's also people on Zoom. So if anyone on Zoom has a question, feel free to put it in the chat.
About the larvicide applied to the inaccessible areas, are they inaccessible because of legal requirements or because of just the physical swampiness?
Yeah, so I think you're referring to how Katie was talking about how for Tripoli, larvicides aren't effective. And so what it is the mosquito that is the main vector for Eastern. It basically is living underneath the roots of the trees and cavities that are formed underneath the root. And when you spray a chemical, you just cannot get into those areas. You can't deliver the chemical to that area of the wetland. We spray these areas every spring for other mosquitoes or some of these areas and I've actually put out little cards to see if I can't see what we're getting into these areas, and we're just not delivering any kind of chemical.
What technology is that? Helicopter spray? Trucks?
Oh yeah, we have. Well, it could be a combination of things, but we have our own airplane. The airplane, it only sprays for the larvae. It's not capable of spraying for adult mosquitoes. So we spray with the airplane.
So if you could get down lower below the lower canopy and spray, that would be better.
No, it's just you're literally, so the mosquitoes underneath the roots of the trees. And so the opening might be this big, it might be this big, and the cavity could be this big. So you have to get the product to travel sideways, right, into the hole that's on the side of a tree. And you have to get enough chemical to kill mosquitoes in this area through a hole that's this big. So that's the problem. Does that make sense?
I understand. Thank you.
Yeah. Uh-huh.
Hi, thank you. Pine Dubois with the Jones River Watershed Association. And, I have a lot of respect for mosquito control, and all the three of you up there have known each other for a long time, but I still have questions. And primarily it has to do with the work that we're doing in the Jones River, which are restoring the aquatic species, especially the American eel, which loves those little crypts. And the tinier it is, the better. And the more mosquitoes it can find, the more it's going to go after them. So I would like to encourage more restoration activity. I think that working together, we can accomplish a whole lot more. And it's a lot cheaper because the more eels we have, the better. And they don't cost anything to allow them to live. But spraying anvil that has PBO in it, when the science says it's bad for the aquatic organisms, that the PBO makes the pesticides last longer, that when it gets in the mud, the sunlight doesn't, make the half-life escalate. We have seen a lot of death in the Jones River because of more because of the aerial spraying when those emergencies happen. I don't think there should be any allowed over rivers like the Jones River, which is a cold water fishery. You're doing, with the fish in there will eat every mosquito in there, as long as we can keep the fish there. And we are basically slowly bringing them back. So I would encourage that. So this science, from Oregon, says really bad for them, really bad for the amphibians too, and will kill, frogs in the larval stage. So you're going to be killing the eels, and you can't do that, And I'm not going to wait for EPA to get around to loving the eel. I can't even get NIMS to register it as a special concern, which it is. If we have to bring them back alone, we're going to bring them back alone. But it puts me in opposition to your program. And I don't want to be there because I understand what you're trying to accomplish. And I believe in the work that you're doing. The work around the tires is unbelievable. The work, when I called the program to help us with unregulated private development, with stormwater systems that were not being managed, you guys are all over it. I can't thank you enough because we are our problem here. If we could ramp up the education so that municipalities weren't creating those kind of, habitats with cattails growing in them and everything, we would be able to spend your money more wisely so that everybody is healthier. And I think the aerial spraying is also just pushing the birds. Every time we have that, pushing the birds north, so they're out of our system, but they're spreading. And I think we're at fault for that. So I would ask, you to look into the anvil, the PBO, see if we can't change what we're using. And until we do, let's get it out of those river systems. If there's a reason to use a backpack in those, I put 11,000 eels in Silver Lake last year, tiny. We're going to be doing this. Work with us. We'll give you a package of eels.
Pine, I just want to return the kind words and say that, we all have known you for a very long time and really respect the work that you do as well, and appreciate that you always ask questions, right? Like that's what the point of this is, and I can tell you that we work very hard with the Department of Agricultural Resources and Fish and Game, and we do, when the area that is going to be targeted for an aerial spray is mapped out, we have setbacks wherever we possibly can that we think that it is not going to compromise human health. And so, and I remember helping with some of those maps where we actually were like, we're gonna stay away from parts of the Jones River, right? So I can't promise you that we can do everything that you want, but I can absolutely assure you that we are taking those concerns into consideration when we make decisions. And we, like, it's a joint process with a lot of state agencies involved and a lot of experts. We take it into account.
I appreciate that, and I believe that I know that because the August aerial spraying, I went off the deep end and put a long message to the governor as well as you and everybody else under the sun, because it was a good year for us in terms of fish return, and we couldn't afford to lose it, and we did not have, because of the work that these guys do, daily. I had people coming to me saying, we don't even have mosquitoes. What are they spraying for? So we weren't the hot spot. So I was glad. The problem I had was nobody would affirm where the spraying actually took place, and I got a real problem with that. Thank you.
I would like to say something as well about the PBO. One of the things about PBO that we at least noticed in testing is that, again, a lot of the recommendations that kind of come with a label are based on that current suite of testing, right? And so if these things, like you said, the impact on, you've got that study from, I think you said Oregon, what you need is just like more data. So like, again, with the neonics, we had suspected for a while that they were impacting bees, but until that data came out of academia, you couldn't just go on a hunch and say like, oh, this is bad, right? You needed that data to really kind of motivate and that's what kind of started the whole, necessitated that need for additional testing, right? And so currently, when you look at the data on PBO, the ecotox data, what you'll find is that the organisms that they're looking at, like things like minnows or daphnids and things like that, it's not really impacting them on their own. So you need some data coming out of it, like I said, like maybe academia or a different direction, and you need enough of it that can say like, hey, look, we need to take another look at this product through a different lens, right? Through a different way, right? And so just a recommendation that, with your organization, maybe that's something that could happen, or maybe you could do something like that. And maybe, again, the more information you put out there, the more likely it is that someone is going to step in and do something about it. Yeah.
Thank you all for joining us tonight. I really appreciate it. I just had a few questions all the way sort of back to the epidemiology of Tripoli. You mentioned the theory about migratory birds being a vector for the virus. Are there any particular species of birds or species of mosquitoes that are vectors for Tripoli?
Yeah, so there's been a lot of studies about that, and it's mainly carried in the passerines. So what we find is that antibody rates are more prevalent in our permanent residents. So like robins, cardinals, chickadees, those kinds of birds. And then you'll see in the summer residents, you'll see antibody rates, and you'll see antibodies also in the summer residents as well. But in the permanent residents, you can have very high antibody rates, maybe as high as like 80%, 70, 80% upward, those kinds of rates. So yeah, they are, those are the main birds that are getting exposed. There's some isolations with other birds as well, but it's mainly the passerines that we believe are driving the cycle.
Okay. So you mentioned residents.
Yeah, like so passerines are
It's a big group of birds. I know what
yeah,
passerines
exactly.
are, but
yeah, so it's all.
but if the antibodies are in the residents, wouldn't that? I guess I'm having trouble understanding where the migrant species fit into that, other than bringing the virus to the residents. And since the residents are resident, if it's in their habitat, they're obviously going to be building up the antibodies. So I understand it.
Well, that's exactly what's happening. So we know from genetic testing that there is genetic exchange between us and Florida.
Mhm.
So we can track that. So we know that the migratory birds are bringing the virus upwards, up north. And so what happens is, the migratory birds would come up with the virus, and then, it gets transmitted amongst all the birds,
Yep.
But the reason why the permanent residents are ending up with higher antibody rates is because they're permanent, right? Like you start to have, you might still have, warblers, say, in your forest, but the warblers there, say, in late August, aren't the warblers that were there all summer, are they? Right? Those are the birds that were from further north.
Mhm.
But the permanent residents are there all the time, so they have an exposure through the whole mosquito cycle.
Yep.
Whereas the summer residents, yeah, they're migrating, and so they're not always the same. Does that make sense?
Yeah, I get that.
Okay.
So is there a reservoir species other than birds? Like I'm trying to understand like how we know that it's originating in Florida and the birds are bringing it from Florida to Massachusetts.
you know, I am not a laboratory person, but it's all about the genetics of the virus,
Yeah.
right? So it's really about isolating the virus from the different species that it's in. And when we're here in Massachusetts, it's much easier to get the virus from mosquitoes and from our human or veterinary cases as opposed to from the birds.
Mhm.
But you know, the birds are infecting the mosquitoes who are then infecting the humans, right? So even if all we're doing is getting the genetics from the virus in the humans, we know it was originally in the birds. And then people in Florida are doing the same thing and putting it all together in a,
Okay.
you know, a genetic tree, right? So that's how we know. I will just full confession. This is sort of like, we know that happens. There is also some thinking that the virus may overwinter somehow, sometimes in Massachusetts,
Yeah.
but exactly how or
Totally.
in what that happens remains to be elucidated.
Sure. Okay. Thank you.
I do want to say bring up one study. This might help a little bit. It's kind of why we kind of thought about Florida, is that you can actually do some like sentinel, you can put out sentinel birds. And what they'll find is like kind of in the panhandle of Florida, the virus just kind of stays there all the time.
Mhm.
So we know that it spends the winter, at least in Florida, all the time, right?
Gotcha.
And then other parts of the country, you can put out the same sentinels and you don't see that.
Okay. Thank you.
I know, I appreciate you, Dinius, bringing up, my name's Kirsten, I don't know if that is important, but about the research and all that stuff, but I got a little lost because I'm not a science person. Are you saying that the research is showing that the amount that's been put out is not impacting as many creatures as we think?
So I think that I'm understanding your question. You're asking if the aerial spray.
I guess my biggest concern is two things. One, what's happening with everything receiving this pesticide? And two, what about the creatures that live on the insects? I mean, that's a food source for bats, for birds, is my understanding, apparently eels.
Yep. Yeah. So it's pretty complicated, and there's actually a lot of kind of unknowns that kind of crop up. But the general kind of gist of what I'm trying to get across is that it's not all like 100% bad, but not all just completely okay. There's a lot of unknowns. So when you conduct this aerial spray, the big focus for a lot of people in kind of like the environmental kind of world is like, how does it impact bees? And so that particular poster that I brought up was kind of showing the impact on bees. Now, like I said, for instance, some of the unknowns are what kind of effect does this have on night flying midges that might be closely related to mosquitoes? Now that is a complete unknown because they're about the same size as a mosquito. They're flying at night. They're not an organism that people look a lot at. And so I don't know. So I can't say again, it's completely doing nothing. But what you can say is that, like, at least for some of the things that a lot of people are concerned about, and what the things that we're looking at. So like when you run that, like I said, you run those tests, you run the tests on bees, you're looking to see how it's going to impact the bees, but you don't necessarily know how that, or even that test, for instance, a lot of the testing was run on Apis mellifera, the honeybee, the European honeybee. And so that's why now they're looking at Osmia lignaria, the blue orchard bee, which is a solitary bee. We looked at Bombus impatiens, which is a bumblebee. And so you're looking at different things, right? Because you want to know if it's impacting things differently, right? But there are millions of insects that you don't necessarily know how that's going to impact them all the same. You can just make the best judgment with that information that you have and based on the sensitivity and how it breaks down, right? So that's at least what I would want to say on that.
I have something to add on that. I want to say it was in 2007, I think maybe. It was early on in when we began doing the adulticiding, the aerial sprays, the state did an assessment of the impact of that we were having on the non-targets. It's been a while since I read that paper. Do you remember the report? I don't. The results? I looked to try to find it and refresh my memory, but I don't remember. I remember them saying that that they did decrease some of the non-targets, but they didn't feel it was significant. But does the lady, is the other person from the Department of Ag, are they on board? Yeah, maybe they have better knowledge of that paper. They might have something to add here. Yes, I don't remember the details. I just don't. But they did look at it. I know they did.
Kim, you should be able to talk.
Hi, my name is Kim Skirm. I'm with the Department of Ag in the apiary program. Can you guys hear me?
Yes, we can hear you.
Oh, you hear me? Oh, okay, great. Sorry, there's a little bit of lag. Also, just a quick note, the chat box is disabled, so I don't know if folks are trying to put questions in it. Sorry, I can't be with y'all in person tonight, but I really appreciate the opportunity to be here. I was hired with the department in 2015, and I'm not aware of the report from 2007, but now that you've mentioned it, I've made note. I'll definitely look it up and we can figure out where a copy of this has landed. I work with the apiary program, and we monitor honeybee hives during the spray. So if there are any questions about honeybee impacts, we'd be happy to answer those. We've done assessments here for the last couple of years with the aerial sprays. So yeah, happy to be with y'all virtually tonight. Thanks for letting me be unmuted.
Okay, I know I got a honeybee guy behind me, so I'll be quick. So basically, anything that's active at night is going to be impacted or going to receive the droplets. So I'm thinking of moths.
Yeah, we don't know. And so, like, again, when you're looking at these studies, like you're trying to make the best judgment.
Yeah.
And you can't test everything.
Okay. And then do we know of any places that control mosquitoes in another manner that's doesn't involve pesticides? And that I'm going to walk and listen.
Yeah, there's Michigan has been dealing with a similar situation, and they also have been doing the aerial adulticiding. So with Tripoli, we're just really extremely limited as to the control approaches that we're able to take. So it's pretty much change people's behavior, through education, outreach, closing ball fields, or it's adulticiding, unfortunately. So that's just the boat we're in. But yeah, like I said, there was a paper the state did roughly about 2007, and I believe they did see some non-target impact, but they didn't feel that it was going to be significant or long-lasting. That was the conclusion I remember.
I just wanted to say one more thing, which is that, there are other mosquito control kind of technologies, right? But the technology that you can use depends on the species of mosquito that you're trying to control, and sometimes the disease and the environment that you're in, right? So you might have heard that there are some places, like I think Florida is using mosquitoes that have been, male mosquitoes that have been sterilized so that you can, in theory, help reduce the population of mosquitoes, right? So the females mate, they don't, produce any offspring. And so you can, in theory, reduce the population. But that, I mean, one, that's experimental. That might have environmental impacts as well. So that's still being evaluated. But again, I mean, the tools that you can bring to bear in any particular situation really depends on the species of mosquitoes that you're dealing with and the habitat that they are in.
Good evening. Keith McDonald. You have an MDR registered apiary. I'm on the MDR do not spray list. I did renew. I'm curious if there is a way from the time frame of a positive detection to a decision point being made that they're going to implement truck-based spraying or aerial spraying, if there's a way to more proactively notify the apiaries that they need to protect their apiaries.
This is it's
Hi, Kim.
Sorry, Kim, you have to unmute again.
it's a great question. This is something that we've really struggled with since doing these sprays because it tends to be really hard. I'm sure Katie can share with you, sometimes just getting the plane, making sure the conditions are correct, making sure everything is ready to go. You know, it's hard. I mean, even with ourselves, we have struggled in the past trying to get beekeepers' sites prepared in advance because we do monitoring before the spray, after the spray, and then a longer period of time after spray, and then the following season as well to check in with the beekeepers. So I do think it's worth, and I really appreciate the comment because we do need to be reminded that as fast a notification is always the most important, and especially if folks are wanting to do preparations to move their bees or at least be mindful of their negative impacts, you know, when a spray may occur. I can let you know from what we've been doing the last couple of sprays, you know, since 2019 with beekeepers, we don't have definitive data on this. We haven't tested it or anything like that, but everything we've seen in as many colonies as we've monitored, there doesn't seem to be any sort of impact if you don't take preparations versus doing something proactively, like putting a cover over your hive, or, I mean, obviously if you move bees out of the spray zone, that's a totally different level of impact. But we've oftentimes, unfortunately, found when folks try to do things to sort of protect their bees, they can sometimes go a little bit too far in the best intentions and may cause further issues. So, you know, if you've got something in mind that you'd like to do to prepare your hives before spray, we'd be happy to walk you through it. We'll be happy to, you know, look at photos, be on FaceTime with you, whatever you need. But I really do, again, I appreciate the comment about wanting to know and more advanced notification. And I wish there was something we could do better with this. There are times when I know there's a green light, and even there are sometimes when those planes can't go up because the conditions just aren't correct. So it becomes a real struggle. There's a lot of variables involved in one of these sprays.
Thank you.
Good evening. Excellent presentation and excellent questions. And thank goodness for the balance between folks like Pine and scientists and entomologists like you. I've had the privilege of going to Syngenta's lab and seeing the research firsthand. They had every soil in the world in tiny vats or whatever, and with radioisotopes measuring all this, you know, incredible science. It does take a long time. And I'm encouraged by seeing newer products coming that hopefully will address all these issues. I know, for example, in golf and in lawn care, the imidacloprid issue with the bees and so forth is being addressed. And they have come up with new products now that takes this, that they can target the specific insect that they're going for and it won't affect the ones around it. So let's hope that I've been chat GPTing over here a little bit about mosquitoes, and there's nothing right on the near horizon, but hopefully there will be, because I think that could help as well. I'm on the trails committee in town, and last Wednesday we had a meeting, and they were talking about some trees that were blocking streams and that we're not allowed to touch them. And I said, "What do you mean we can't touch them? They blew down. I can't go and safely remove them." And they said no. And is that a state law, or is that a conservation? Or because I just saw this beautiful slide up there of you removing trees, and I'd be right out there now doing it if I could. And I think there's some things we can do immediately in the short term to try to maintain the streams and keep things moving. And so I was a little frustrated when I heard that we can't remove trees from streams. And another quick, before I forget too, the question was asked why they couldn't identify where their airplane had sprayed. I believe that's all GPS when they spray, correct? When they click that sprayer on, it should be very accurately. So is that information available for the person that had asked for that? Right. Because I've been in helicopters before that were spreading fertilizer, and it was it was very precise. They call it precision agriculture, and I would think those records would be available to see exactly where the sprays took place. Thank you.
Yeah, I mean, the state, the planes do have trackers on them and you can follow the application. Of course, you can't see on there whether the sprayer is on or off. What they're doing is they would fly, if there's an exclusion, they will fly it and they just turn the sprayer off. So you wouldn't necessarily know, the state does get that data, but it's not publicly available.
When I flew in the helicopter as a co-pilot, they had GPSed it first. They did the perimeter, and then when they clicked on the machine, this happened to be a fertilizer spreader, but it's no different than when he sprays. It showed it paints a line exactly when the sprayer was turned on, and then it shuts that actual paint line, I call it, on the GPS in front of the big screen in front of them. So we can paint that line and fill it in and be very, very, it's very precise, because I also stood on the ground to make sure that it stopped exactly where I wanted to and it landed at my feet. So it's called precision agriculture, and they're getting better. I know that Fred's working on drone technology to try to minimize impacts as well. So there's a lot of promising things coming. Sometimes it gets tied up, which for good reasons a lot of times. But anyway, my 2 minutes is probably up, but thank you.
You're welcome. Yeah, those planes do have that technology, and our own aircraft has that technology. So they basically are provided with maps, and they know very precisely the spray block and where there's to turn the sprayer off and turn it on. There is starting to be a little bit more automation with that, like to be able to automate it so that the sprayer it will shut on and off automatically in the spray block. Our airplane doesn't have that ability. I don't know if the contractor has that ability yet, but yeah, it's getting ever more precise, and they very carefully characterize the aircraft to make sure that it's spraying at the proper rate and the proper droplet size. Droplet size is extremely important. If the droplets are too big, they fall out of the air too fast, and you would have a greater non-target effect. And if they're too small, then they'll float, they'll go somewhere else. So yeah, those kinds of things are monitored very carefully, spray conditions and mapping and stuff.
And can anyone answer my question about the streams? I'd love to get those.
So Sarah works for Department of Energy and Environment. So Sarah, if you could just touch on that briefly.
Yeah, thanks for that question, Chuck. I can't speak to any regulations. I'm still learning a lot in my role. But I will say that I do know that having brush in the streams like that is good fish habitat. I took part in a land stewardship workshop as recent as last week, and that was one of the recommended practices to actually let trees fall into some streams to provide better habitat for fish. So I can't say for certain about a regulation, but I certainly could check with my coworkers and get back to you. They have a wealth of knowledge on the regulations in our department. So I will check on that and I will reach out to you. Just make sure I get your contact information after this.
Okay, great.
I would imagine that it's just to better support the fish.
I think instead of having a regulation that says you cannot remove a tree from a stream, it should be more common sense, like, okay, we want to build fish habitat. Let's go over here so that we can still have some, it could create a little eddy or whatever. But, regulations get tough because if you can't remove trees, we're going to have more mosquitoes.
Well, I think some of it has to do too with the liability of volunteers with chainsaws.
Yeah,
on public
I understand.
property.
Yeah.
And then also if there's like conservation restrictions and things like that. But so Patrick, it looks like you might want to chime in.
Good evening, everyone. My name is Patrick Farr. I'm the Assistant Conservation Agent for the town. So, in response to your question, um, the Conservation Commission doesn't say no a lot. So, I don't know who told you that you can't do it. What you have to do is you have to apply for a permit. It's called a notice of intent. So, through the state's Wetlands Protection Act, 310 CMR, and the local bylaw, Chapter 196. The applicant can request to do whatever the type of activity you'd like to do, but you have to do it in an environmentally responsible manner, and you have to provide a reasonable justification for that type of activity and provide either avoid, minimize, or mitigate. So, yeah, again, it would be a filing of a notice of intent for that type of activity. Sure. Go and ask this question.
I was just saying, does that happen all the time, or is it on public land? Is it on private land? A private homeowner, I imagine, could remove it unless it goes onto the land of another. I know there's a lot of rules depending on whether it leaves the property or not. And I don't want to get bogged down on this. We can talk about this another time, you know.
Yeah, I'm happy to speak to you offline on this. Okay, thanks.
So we probably have time for like one more question.
Two things. Is there a protocol? I'm sorry. Two things. Is there a protocol for how to notify the public that, hey, this is going to happen, close your windows, come inside, there's going to be spraying? That would be number one. And number two, have you noticed that the infected bugs tend to be in a certain area, like near these swamps?
So I can definitely talk to the protocol about notifications. So when an aerial spray operation is going to take place, there's a joint press release that comes out from the Department of Public Health and the Department of Agricultural Resources. We also hold multiple phone calls with environmental interest holders, with the legislative groups who are involved and the local boards of health, and sometimes also with municipal leaders. So we do as broad notification as is possible in a relatively short amount of time. And information about, people who are concerned about possible exposure to the spray, and therefore, they can close their windows and put their air conditioner on, recirculate. That is part of the messaging that goes up. And there's also a fairly robust website that has information about, protecting bees and swimming pools and, outdoor pets and, air conditioners and things like that. So yeah, so we have a pretty extensive outreach. Ellen is probably the right person to talk about where the mosquitoes are, but that's why everybody does surveillance, right? Is so that we can figure out where the populations of mosquitoes are largest that particular year at that moment, and which ones are, carrying more virus. And that's all part of the data that is used to help make decisions about control locations.
Yeah, I would just add that we do know that mosquitoes move around more than you would think from wetlands. It depends. It's one of those things where it's species specific. So there were actually mark and recapture studies on mosquitoes, believe it or not. They dusted a bunch of mosquitoes, and then they put out a bunch of traps and they recaught those mosquitoes. This is for Melanura, the main vector for Tripoli. They recaught them 5 miles away from the wetland. So we know that these mosquitoes move around much more than you would expect. There are other mosquitoes, particularly the container mosquitoes, where they can be very local to their place of origin. But unfortunately, with Tripoli, that's not the case.
Okay, so we only have a couple minutes left. I know there was one last question, so. 2 minutes.
I actually have 3 questions. Meg Sheehan from Community Land and Water Coalition. How many years have you been spraying? Decades? I mean, I remember it from when I was a kid. No. The trucks. DDT. I mean, how many years has this been going on? 50?
You mean for the
How
aerial
many years have you
delta
been aerial spraying for mosquitoes in southeastern Mass?
So if you're talking about aerial spraying alone, there was a spray in 1990 and then not another one done until 2006. And then, as I tried to show on my graph, there have been multiple, unfortunately, we've seen kind of a proliferation of years with activity. And so there have now been multiple sprays since then. And don't quote me, 2006, 2010, 2012, 2019, and 2024.
None before 1990? You never?
I don't know.
Oh, because
I'm
I
not
would say
that
for
old.
sure. Well, I am. Anyway, also another question. Are you spraying all open water? All over all water bodies or none or?
They're not. They're not. The spray doesn't occur over drinking water supplies. I don't know about if they're not drinking water.
No, it's just.
Yeah, they're not sprayed over drinking water. It is sprayed over,
Ponds.
Yeah.
Yeah, I mean, I can attest that last summer, I went to our pond after you sprayed and it was dead. There were no dragonflies, no nothing. Whereas the day before it was a vibrant, insect activity site. So I really think that all water bodies should be off limits and you shouldn't be depending on homeowners to notify you every year to get off the no spray zone. And, this is our sole source aquifer, 200 square miles. So when you're spraying on the water body, you are spraying the aquifer. It's an expression of the aquifer. So I really think you need to take that into account. And then my third point is
I'm sorry, can I just respond to that for a moment? Because there have been studies done in Massachusetts after aerial sprays doing water sampling to detect both the active ingredient in Anvil 10 plus 10 and PBO. And no Anvil 10 plus, no sumethrin was found, which is the active ingredient. And I believe that the PBO detection was only in one location. So it's really unlikely that there are significant amounts getting from the water into the aquifer.
Well, the water is the aquifer. So if it's in the water,
Right,
it's
but
in the
if you
aquifer.
take a sample the next morning,
Yeah.
the morning immediately afterwards, and you can't detect the product,
So it disappears immediately.
exactly.
Well, I guess then my third question is maybe moot, but have you ever looked at the synergistic effects of all the, the spraying plus the industrial scale agriculture that we have here in the form of the cranberry bogs, that aerial spray, pesticides, fertilizers, etc.? Has anyone looked at, are you spraying over the cranberry bogs too? You're spraying everything. Okay.
So as far as I know, there hasn't been any like research specific to Plymouth County on that, but just in general, I think that a lot of what fingers are kind of pointing towards, we talk about things like a lot of things like insect declines, and we're really focused on kind of the impact on wildlife and what's happening. And a lot of it kind of comes down to this confluence of factors. So when you're bringing up this point on the spraying and then the industrial agriculture, but there's also other components that kind of go into that as well, like the introduction of invasive species, you have climate change, and really kind of the big thing that kind of comes up the most out of any of these factors that scientists in general kind of agree on is the deterioration and loss of habitat is really the big one. So if you were, for instance, to put up a new housing development, that would have a greater impact on your wildlife in an area than an aerial spray would.
But,
And
but
so,
that's not,
yep,
sorry.
you can go ahead. I didn't mean to
We're
interrupt.
talking about chemicals, not about land use change. We're talking about cumulative volume of chemicals being dumped on our wetlands and our waterways year after year after year.
Okay, so I can talk on that then. So, kind of going back to the ecotox testing, right? A lot of they do look at things like that. So how does this product break down over time, and how long does it take to break down? As an example, like Katie brought up that study where you can test drinking water and you can not find any sumetharin in that drinking water. That is not surprising to me, just because we look at that stuff in the whole fate and metabolism department. You're looking at that. So one of the crazy things is that they will actually go and they will look at sediment, they'll look at water, and they'll look at very, very small trace amounts of this. Like we run instrumentation that can detect less than a drop in an Olympic-sized swimming pool. If it's there, it's there. And if you don't see it, it's not, it's like physically not there. So when you pick up a cup, you're looking at the molecules that are there, and they break down. So
I mean, I hear what you're saying, but
Yep.
then you're also saying we don't know the impact. But the real question is the synergistic effect of all the different chemicals together. Do you have a study on that?
No.
All right.
Okay, we're at
Mhm.
8:03, so I want to be
All right.
respectful of everyone's time. I want to thank everyone for their questions. There were a lot of questions, and for our presenters, we covered so many topics. This was really interesting. So before we wrap up, Sarah was just going to talk briefly about the town of Plymouth and the climate action and adaptation plan, as I mentioned, as it relates to vector-borne diseases and climate-related health risks. So Sarah.
Thank you, Andrea, and thank you, Sustainable Plymouth, for organizing this event tonight, and for all of our panelists here for coming and answering everyone's questions. Thank you all for being interested in the environment here in Plymouth. So I'm Sarah McCormick. I am a natural resources and sustainability specialist in Plymouth's Department of Energy and Environment. And I just wanted to take a second to tie this event tonight to the work that we're doing here in town. As some of you already know, in 2022, the Plymouth Select Board declared a climate emergency and established the Climate Action Net Zero, or CANS Committee, to construct the town's first climate action and adaptation plan. CANS, along with town staff, worked to craft a plan to help the Plymouth community respond to climate-related threats and opportunities and prepare to meet net zero commitments. I'm happy to report that this plan was passed this last spring, and we've hit the ground running to implement it here in town. Through the Climate Action Plan, Plymouth is adopting a whole-of-government approach to combating climate change. The plan includes 62 action items over 5 main focus areas, and those focus areas are buildings, energy and infrastructure, health, safety and preparedness, natural systems, transportation, and waste. The first goal of the health, safety, and preparedness section, and I will read this right out of this book that I'm pretty much carrying with me at all times. So the first goal is to ensure that all community members are prepared for and have the tools necessary to endure the impacts of a changing climate. And we know that environmental changes associated with climate change are expected to result in higher disease burden within the community. Increasing precipitation means that there will be more standing water. Conversely, droughts might mean adapting or altering agricultural methods, which might bring on a slew of other issues. Increased temperatures mean longer transmission seasons. We know that first frosts are coming later and last frosts are occurring sooner. These changes, among others, are expected to extend the range for vectors like mosquitoes to areas previously unsuitable, and increasing the risk for certain members of our population. So action HSP, Health, Safety, Preparedness, 1.2.B, again, I'll read this right out of the Climate Action Plan. It says that we will partner with local and regional health and emergency services to track and report on vector-borne diseases and climate-related health incidents, and provide comprehensive training on climate-related health risks, particularly among vulnerable populations. turn my little cheat sheet over here. So as we implement the climate action plan here in Plymouth, we are going to continue to offer opportunities and pair with community organizations like Sustainable Plymouth to be able to conduct outreach and engagement and be able to get the best information to those residents here in Plymouth. That way we can mitigate the risk of climate change together. Before I go, just a few announcements. We are really fortunate to work with a group of incredible, dedicated volunteers, and we couldn't do this work here in Plymouth without them. Many of them are in this room tonight, so thank you all very much. The CANS Committee, again, Climate Action Net Zero Committee, which focuses on those 5 focus areas. They have 1 vacancy right now, and they're accepting new members to working groups at all times. So if you're interested in all joining or becoming involved in implementation of a climate action plan, please see me after this meeting. I will give you a card, or you can also visit our division, the Division of Sustainability and Resilience site online. Also, Elaine very kindly reminded me to let you know that we have all these wonderful flyers up here. We'll be hanging around for a few minutes after to talk you through some of them. One of them is information about the Code Red emergency notification system, which is a county-wide system that is used in order to send emergency alerts to the public. I will say I'm enrolled in it. They're very limited on the communications. They only send out important stuff. You are not spammed. Happy to walk you through that if you're looking to enroll. We are partnering with the Sheriff's Office on that as part of the Health Safety Preparedness section. And then we are also hosting a community workshop this Saturday at 9 o'clock in the 1749 courthouse. So not this courthouse, the other older courthouse in town. That is going to be focused on our downtown resiliency project, which is aimed at reducing the risk of, or the effects of the urban heat island, which is sort of the downtown area here in Plymouth. So if you're interested in hearing what we have planned and want to provide input, again, that's 9 a.m. at the 1749 courthouse on Saturday. Thank you guys all so much for being here. We really appreciate it.
Yes, thank you so much to Katie, Ellen, and Blake. Thank you everyone for coming. Have a good night.